Schizophrenia, which is a mental illness that causes its victims to hallucinate or become delusional, is historically one of the most difficult illnesses to treat. In addition to being difficult to treat, schizophrenia has no known cure, and its initial cause is also largely a debate. Many believe schizophrenia begins to develop at a very early age, with genetic factors being the probable cause. Others believe schizophrenia occurs when neurons in our brain become dysfunctional. Nonetheless, schizophrenia is a common illness, as it affects approximately 1% of the total global population. While studying the brain, James Maximitz, Jeffrey Kahn, and other researchers at Vanderbilt University discovered that a particular protein in our nervous system, mGlu1, can be targeted for treatment of schizophrenia. mGlu1, which is short for metabotropic glutamate receptor subtype 1, can be drugged to enhance its functionality, which causes a reversal of some common symptoms of schizophrenia, specifically cognitive deficits. According to the study, cognitive deficits include impairments in working memory and attention. Despite there already existing some treatment options for schizophrenia, these treatments target the positive symptoms of schizophrenia, which are delusions, hallucinations, and disorganized speech, and leave the negative symptoms of the illness unaffected, which are flattened affect, reduced speech, and lack of initiative. These researchers discovered that specific positive allosteric modulators, or PAMs for short, which are basically a group of substances that can boost the efficacy of a receptor, can be combined with mGlu1 to reverse those negative symptoms that are associated with the illness. To test this hypothesis, researchers experimented with lab mice. After allowing the mice to settle and become acclimated to the testing facility for a week, they were placed in a Y maze and were studied as they completed miscellaneous tasks, some with a source of light and others with no light. During the experiment, their spatial working memory was actively monitored. From this experiment, these researchers found that mGlu1 PAMs can reverse cortical hyperactivity and have efficacy in reversing working memory deficits. This method of targeting mGlu1 with PAMs ultimately poses an efficient method of treating schizophrenia by restoring the ability of dysfunctional neurons, which is thought to be the initial cause of schizophrenia.